Okay, good morning, everybody. I think without much ado, we should get started with the first session. Just a suggestion for all the speakers, you please try to maintain the timing. There will be one bell which will be rung at 12 minutes, followed by which after 3 minutes, there will be a final bell. So try to wrap up your presentation within that. Try to finish early so that we will have scope for some questions, right? Okay, so I will call upon the first speaker today, Dr. Tejveer Singh. He's going to speak on anti-cancer and anti-HPV activity of green synthesized silver nanoparticles doped with berberis extracts.
Thank you, sir, inviting me. And good morning, everyone. Myself, Tejveer Singh. First, going to presenting my work, I would like to introduce myself. I did my graduation from University of Rajasthan and masters from University of Hyderabad. And in 2021, I have completed my PhD in Delhi University. And I got the DHR Young Scientist Fellowship and currently working in JNU. And day before yesterday, I joined as an assistant professor in Delhi University, Ansraj College. So today I am going to present my work on the cervical cancer and what is the cervical cancer. So cervical cancer is the fourth most common cancer worldwide. The table here this table shown here depicts the annual incident and annual mortality of the cervical cancer in worldwide as well as in India. And this is the columnar junction where the HPV persistent infection of the HPV lead to the cervical cancer. Uh, these infections lead to approx 7 to 10 years to convert normal to malignancy by the HPV. So the main cause of the cervical cancer is HPV. HPV is a human papilloma virus, that is the double stranded DNA virus, which accounts the genome size 8 kb, and there till date there are the more than 200 type of the HPV. There are the two category. First one is high risk and low risk. High risk, low risk means they are the high probability to cause cervical cancer. That account HPV 16 and 18. So to eliminating or approaching the treatment of the cervical cancer, today we have the different type of the treatment. First is the vaccine, prophylactic and therapeutic vaccine, and chimeric vaccine. Chimeric vaccine is under development. So after that, the HPV elimination, we use the different phytochemical formulation, Basant and Pernim. Basant is formulated by the Professor Talwar, and these the different anti-HPV therapeutic which going to lead suppression of the antiviral activity by the berberin, curcumin, and Embolica. So these the phytochemical which have already seen to eliminate HPV and treating the cervical cancer. These the low grade with the curcumin, pernim, basant. These are the eliminating of HPV with the infected cervical cancer lesion. And curcumin, berberine, Embolica. These are the inhibition of the by down regulating the transcription factors such as STAT3, AP1, and NF-kappa B. These are the different transcription factors which lead to cause cervical cancer upregulation and downregulation. These HPV lead to the upregulation of the STAT3, NF-kappa B, and AP1. So we can downregulate these transcription factor by using these different phytochemical. There are the three type of phytochemical. First one is the pure compound like curcumin, berberine, and other phytochemical are the polyherbal formulation like Basant and Pernim. So before going to that treatment of the cervical cancer, so I mentioned that these phytochemical already well established to treat cervical cancer, but they are not coming in the treatment. So what is the lacking? What is the bottleneck? So that is the pure economy interest from the pharma company. Clinical trial are emerging to be more and more expensive, and labor extreme demand exhausting infrastructure. So what is the alternative? So we are going for alternative. So there are the two approach, allopathy and Ayurveda, Ministry of Ayush, under Ministry of Ayush. So we are going for homeopathy because homeopathy have second line in the all over the world. So these are the cost effective, huge herbal formulation, and these are the less side effect with high efficacy. So now I would mention the key question: what are the homeopathy and how they are work with the anti-HPV, and what is the established formulation. So these are the list which have our parental lab already working on these phytochemical, curcumin, basanprini, and so on. So these are the already homeopathic formulation plant drive which have already used for the treatment of the cancer and cancer lesion and what Thuja, curcumin, hydrated. Those are the highlighted which I used for my study. These are the homeopathic formulation, the origin, plant kingdom, and in kingdom pure compound. So out of these I choose plant derived formulation. There are the two types, mother teacher and potency. Mother teacher means in homeopathy preparation they use pure extract of the drug, and potentization means they use the extract of or dilution of the mother teacher. These are the scale, C scale and X scale. X scale means 1:10 ratio. One part of the original extract and rest 9 part of the bical or alcohol or whatever they are used soluble. And C means they use 1:100 dilution. One part of the drug and rest 99 part of the alcohol or water. So I used these 30C and 6C for the drug treatment dilution. And the assumption is that approx 10 power 6 Avogadro number. In the 10 power minus 6, there is the only one original molecule from the mother tincture. These are the drugs which we have procured from the Ministry of Homeopathy: Berberis equifolium, Berberis vulgaris, Mentha peperata, Curcuma longa, Cinchona officinalis, Thuja hydratis. So first I checked the cytotoxic effect on the different cell line in vitro. C33A, Cia, HeLa. This is the mother teacher, and with the potency of 30C. This is the dotted line showing the vehicle control, and the dark line showing the effect of. So we can clearly see the effect of the mother tincture in different cell lines. C33 is the cervical cancer cell line, but there is no HPV positivity for control. And CI is the HPV positive, 16 positive, and HeLa is HPV 18 positive. So all these drugs is showing very good effect on that cancer cell line. Further, I evaluate the antioxidant activity by the using this same drug and calculate the IC50 with the available drugs, 5-FU, cisplatin. These are positive control, and these are the how they are homeopathic drug with the correlate antioxidant activity with the different TPSA, ABTSSA, and DPPHSA. Further, I used these phytochemical ingredient by the using docking with the different HPV viral protein 4X, R8, and 6SJ. These are the target of E6P3 transcription factor and zinc motive ion and different. These is the molecular docking. They are the showing the curcuma longa. That is the protein. Green is showing protein, and this is the berberine. These are the conclusion of that paper. All these drugs is used for the antioxidant activity and the cytotoxic effect. Then check the molecular docking. Then further I go the molecular mechanism behind of this. There's the Western blotting of this, the transcription factor and ISC, and these are the expression of the STAT3 and HPV oncoprotein. So these drug is showing well HPV 18 and 16 going to down regulate with the effect of this phytochemical Berberis equifolium. So that I evaluate this. The conclusion: use the Berberis equifolium to control or side effect, less side effect with the low IC50 and cell cycle arrest in G0 and G0 phase. pituitary juvenile expression downregulate. There is a no effect of the Junbi, and further checked by the bioinformatics. This is the future research roadmap. So I am just going to in JNU fabrication of green synthesis of Berberis actifolium coated metallic nanoparticle AGN treatment for HPV specific cervical cancer. So further for study, I use this Berberis actifolium plant extract for preparation nanoparticle. These are my publications. These are my mentor, which I worked in from University of Hyderabad 2012 to 2022.
So you have tested several of these homeopathic formulations, right?
Yes.
So you didn't comment upon which one seems to be better, which one seems to be not doing as good, and whether that has got anything with whether one has a better effect on one cell line compared to the other cell lines. You should have. Can you comment something on that?
Yes, sure, sir. So I just take a slide for that because all homeopathy are showing very good effect, but the difference between the effect of different homeopathy drug, that is the conclusion slide of the Berberis actifolium, Berberis vulgaris, Mentha peperata, Curcuma longa. So double positive showing that they have very good effect at C33, Sia and Hela. Out of these drugs, Berberis actifolium showing very good effect for both cell line with the comparative control. Although Berberis vulgaris also showing, but that is not showing in Sia, and of Thuja is also showing, but that is not showing with the HPV 18 positivity. So we have to control both HPV 16 as well as HPV 18.
Have you combined these homeopathy drugs?
No, ma'am, I have not tried that combination of that drug. But we have consult with the how we choose that drug. So we have to consult with the Ministry of Homeopathy, DG, Dr. Anil Khurana. So they suggest and discuss the which homeopathy you have to use with the pure compound. So that's why we choose this.
What is the exact outcome of this study?
Sorry, ma'am.
What is the exact outcome of the studies? What we should take or what should be recommended? What do you say?
This study conclude that these phytochemical Berberis actifolium have very good effect in vitro on the cervical cancer case. HPV in case of cervical cancer, as well as specific HPV positivity in respect of HPV. So we can downregulate by the controlling HPV oncoprotein like E6 and E7. So we can go further for in vitro study, and in my future roadmap, I used in DHR Young Scientist Fellowship, choose 3D culture, because after the completion of PhD, I gained the experience of the 3D culture also. So I just working on that, how the working with the 3D culture.
Do you recommend it for like how long duration, or is there anything such coming out?
Yes, ma'am, that Berberis actifolium is showing good effect.
Okay, you took this homeopathy medicine, did you take it right from the plant
pure compound
and compared? Yeah.
Yeah, sir, ma'am, I will try for that also with the
Okay.
this
Thank you.
homeopathy formulation. Good afternoon. No, sir. These are the normal. We can develop also the resistance splatin, but after the treatment of chemotherapy, that is the side effect of the chemotherapy. If we are giving cisplatin 5-FU, they will develop the resistance. So that's why we are going for the alternative pathway, phytochemicals such as these, which are mentioned by me. Yes, sir. It's depend on the various culture, how they are working with the patient and the in vitro study. It depends. That is not exact with the human and animal and cell culture. So yes, sir. I in future I will try to do synergetic effect, the combination of the different phytochemical. Thank you. Thank you.
Good morning, everyone. My topic of presentation is dynamics of the conformational transition of biomolecule from a theoretical perspective. We all know that protein are large complex molecules which play a very important and many crucial role in all the living organisms. Here the unfolded protein fold in the standard physiological condition under temperature, pressure, pH into the native state, and after when it is in the native state, it makes conformational change by varying the temperature, pressure, pH, and the mutation in the polypeptide chain, and it attains the misfolded structure. This misfolded structure this misfolded structure may self-associate to form an aggregate, which leads to the several neurodegenerative diseases like Alzheimer's disease, Parkinson disease, and Huntington disease and ALS diseases. Now, this folded protein, misfolded protein, and the aggregated protein may be understood through this energy landscape theory, where the unfolded protein lie at the top of this funnel. This is the rugged type of energy landscape, and the misfolded protein and the native protein found at this global minima. In between these two states, this unfolded state and the native state, there are several non-local minimas are present, which may contain the folding intermediate and the partially folded intermediate, which we call as a misfolded state of the protein. This misfolded protein may self-associate to form an aggregate, and these aggregates are much more stable than the native state. Means if once this amorphous aggregate or this amyloid fibrils are the aggregate forms, it is very difficult to cross the potential energy barrier and reach it to the native state, and it leads to the neurodegenerative disorders. So here, any type of conformational transition in this energy landscape may be modeled in two-dimensional energy landscape, where at local equilibrium, all this multi-dimensional energy landscape is converted into two-dimensional energy landscape. And here we study the transition of misfolded protein, because it is very important. As the protein turnover mechanism fails, it leads to the protein aggregation diseases. So by using molecular dynamics, Langevin dynamics, and generalized Langevin dynamics simulation, we study the conformational transition of amyloid beta, because amyloid beta is the principal component of the amyloid diseases in the Alzheimer patients. So the basic difference between molecular dynamics, Langevin dynamics, and the generalized Langevin dynamics is that in the molecular dynamics, we use this classical Newton's law of equation, but by considering the Brownian motion of the molecule in the solution, we may use this frictional term and this random term also. So this simple Newton's law of second equation is easily converted to this Langevin equation, where this Vt is the instantaneous velocity. Means we calculate the velocity at this point only where the molecule it is. It not calculate the velocity at the past. It did not remember the velocity of the past directions. But if we generate this velocity in the past direction also, this equation we can easily convert it into this generalized Langevin dynamics equation, where we use the time dependent frictional memory kernel, and by using this equation we use this generalized Langevin dynamics simulation. So using these three, this is the simulation protocol of our MD LD and the GLD simulation for the conformational transition of the amyloid beta into the misfolded state of the amyloid beta. From this simulation, we calculate the transmission coefficient, or simply we can say the rate of this transition coefficient by using the reactive flux formalism. I am not going to detail of this formalism, but here in this figure, we can say that from the LD simulation, we can say that at t is equal to 0, initially the transmission coefficient gives the maximum value, which can see that at initial times, there is no transition of the unfold to the misfold transition. But with increasing simulation time, the transmission coefficient decreases at the unfolded protein converted to the folded state. And after a long time, this transmission coefficient reaches to the 0 value. It means it is vanishes after a long time. But it is not true, because some studies say that after a long time, there is some solid reaction is also works. The rate can't go to the 0. So to formulate this, we can use the generalized Langevin dynamics. One's theory is that at longer time, this Langevin dynamics fail because of the at longer time the solvent forces doesn't work in the complex protein solvent interaction. So we use generalized Langevin dynamic simulation study, and here we can see that at very low value of the frictional coefficient, gamma is equal to 0.4 picosecond inverse. The transmission coefficient gives the maximum value, which means that there is no unfold misfold transition at very low value of the frictional coefficient. But with increasing value of the frictional coefficient, the transmission coefficient sharply decreases and attains the constant value, which indicates that the constant value of the rate. But for the higher value of the frictional coefficient, like gamma is equal to 5.0 picosecond inverse and 10.0 picosecond inverse or even more, the transmission coefficients monotonically decreases and did not reach the constant value, which indicates that there is no well-defined rate constant for the higher frictional coefficient and the presence of dynamic disorder in the system. To study the dynamics of the conformational transition of amyloid beta, we calculate the potential of mean force from the simulation trajectory, and we can see that in the energy landscape, we can see that two well-separated minima are present. One denotes the unfolded state and one denotes the misfolded state. Here we can see that the conformational transition of amyloid beta is rugged energy. We found the rugged energy landscape. We can say that the transition from unfolded to the misfolded state is occur in the heterogeneous manner. So to calculate the dynamics of the conformational transition of amyloid beta, we analyze the transition path in between the unfolded and the misfolded state. For this, we can randomly select two boundaries in between this unfolded and the misfolded state. This is XU and this XM. Below the XU, the unfolded proteins are present, and above the XM, the misfolded proteins are present. But in between these two boundaries, the conformation of amyloid beta is present as it enters from one direction and exit from one direction. And by calculating the number of transition in between this XM and XU, means in between the transition path, we can calculate the transition path time distribution. And here we can see that from the LD simulation, initially the transition path time distribution high, which means that initially the misfolded protein converted, initially the unfolded protein converted to the misfolded protein. That's why this distribution is high. But after a certain time, the transition is less, means protein going to misfold, and we get the lower distribution. And after a very long time, this distribution becomes less. It tends to 0, which indicates that all the conformation and the misfolded protein reaches to the all unfolded protein reaches to the misfolded state. Similarly, in GLD simulation, we also get the same trends, and our simulation, we can fit our simulation with some experimental and theoretical results and calculate the energy and the curvature of the protein. Here we can say that energy is very high, which indicates that once it crosses the barrier, it is very difficult to reach the unfolded state. And we also calculate the LD simulation with varying transition path time distribution with varying value of the gamma. higher the value of gamma, higher the interaction, and we get the higher distribution of the protein. We also calculate the mean transition path time. It is the time in between when the conformation of amyloid beta lie in these two boundaries. So here we can see that as the length decreases, this time is decreases, as we can see that from the mean first passage time. And we also calculate the unfolding pathway of the adenine ribose switches from the SMD simulation by fixing this residue, this atom of the, and by constant pooling at 3 prime, and we can study the conformational transition of the adenine ribose switches. This is the unfolding pathway of the ribose switch aptamer, and we get the 5-stage transition from the adenine ribose switch aptamer, as each step is denoted by the rate, initial state is denoted by the rate limiting state, and followed by all the states. So this is the conclusion of our talk. This is the Langevin, and we study the Langevin and the generalized Langevin dynamic simulation and compare with Kramers and Grote-Hines rate theory. And initially at t is equal to 0, there is no transition of the protein from the unfolded state to the folded state. But in GLD simulation, we get the well-defined rate constant, and the transition path time revealed that at low frictional coefficient, the distribution is low. For the high frictional coefficient, the distribution is high. And we also calculate the mean first passage time with varying length of the transition path time. Thank you.
Yeah. So Vishal, I think you have dealt with a subject, which requires a lot of mathematical background. Yeah. So I'm not sure how many people in the audience would understand the equations that you have shown. I'm not saying that what you have done is not good. I mean, it is very good. But maybe you may be knowing the equations, or I may be knowing the mathematics behind it. But what I can only tell is just give a small comment on the presentation in general.
Okay.
So, for example, you used a word called transmission
coefficient.
Transmission
coefficient.
So, what does transmission coefficient?
Transmission coefficient actually gives the rate. Actually, from the transition state theory, this is the till now only one parameter. Means people more studies it's the transition state theory.
So here. So the problem that I want to highlight over here is,
okay,
in order to explain something, you suddenly brought in transition state theory.
I'm coming. Okay.
Right.
From the direct point of view, we can say that it's just the rate.
Break it down into smaller parts.
Okay.
See, at the end of the day, we are not interested in what the mathematical equation is, right? But what your analysis is essentially telling us in terms of what is that question that you have been able to address in the general problem of protein folding and misfolding. So that message was not really very clear.
Okay.
One. And second thing is that, you have published it in ACS Omega, right? So definitely it has to be of that standard, otherwise it will not get published there. So I have no problem with your publication and your work. The question is that when you send this message out that, look, this is what I've done and this is why it makes sense, you have to craft your way through your presentation that is convincing. For example, let's say you showed a lot of graphs and you showed your citation also, but there are other citations as well. So I was, as a listener, was confused. What part of it is being cited for somebody else's article, and what is it your statement relates to your article? So there was a lot of. You were going in one wavelength, but we were not receiving it. So that's what I don't know what.
Similar kind of comments I would like to give. You're doing all these studies, you're only dealing with some software. What is the input?
No,
and
first
what is the
this is
output?
mathematics plus software. First we derive the mathematics. According to we modulate the theory and perform the simulations.
Okay.
Yes. This Newton's law, huh? Newton's law.
Yeah.
Okay, but it takes time, so that's why I just go through the. Means this conformational transition of the biomolecule. The first we study the conformational transition of the protein amyloid beta. After that we study the conformational transition of the riboswitches, which is 5-state transition, which give the more clear picture about, because proteins are mostly the 2-state transition, unfolded or folded, or the folded misfolded, this 2-state transition. But in biomolecule we can see that even in the hairpin also we can see that it's mostly the 3-step process. So determination of the rate is very difficult. Which state we have to choose? So according to this dynamics, we use the rate limiting step to calculate the rate and all these things. Means depending upon the biomolecules, the conformational transition changes, even in the force also. So
Okay, thank you. Okay.
You're trying to implement those into the basically disease biology or any biological models.
yes.
Are you trying that?
Yes. I am first means from the beta amyloid, we simulate this beta amyloid in the misfolded state, and we calculate, and we want to grab the information from the transition state, from how it is going to the misfolded state, or
And it's-
means rate. In the rate key means if the through the rate, if the frictional coefficient is less, then the rate will be slow. When the frictional coefficient high, it going to the means is partly going to the one well to the another well, means the misfolded state. Means it depends upon the rate and the frictional coefficient.
Same is a question about the aptamers you people are making.
Which
Aptamers.
no
No. You mentioned about
aptamers.
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 Yes.
We are not making. We just take the aptamers from the RCSP PDB data bank, and we simulate these aptamers.
So the work stop at simulation, or you go for the further applications or of that or not?
No, first we do the simulation. After that, we go to the applications.
Thank you.
Okay.
So I think I will. So you are ready now? You are ready? So you are yet to upload it over there? Yeah. So let me. Okay. Okay. Fine. Go ahead. Yeah. So Dr. Bindu Alur is going to speak on Rincosia herbs in the treatment of cancers, right?
Good morning, everyone. I'm Dr. Bindu Prasunna. I'm from Andhra Pradesh. I'm DST Human Scientist A there. So my topic of seminar here is Rincosia herbs. Nowadays, we know cancer is very dangerous and dreadful disease, and it needs a lot of attention, and treatment is also very difficult. And cancer has been the leading cause here, and almost 1 in 6 deaths are seen in 2020, and this has been recorded by WHO also. And the main causes of cancer are mutations in form of environmental effects, physical and through chemical carcinogens. Even when we handle this SDS page and other electrophoresis chemicals also, those are also causing this, those are carcinogenic, and in future it will cause cancer. So biological carcinogens are in the form of virus infection. After that, it will develop into cancer. And types of cancer. So carcinoma is from skin, starting from skin, and this sarcoma is connective tissue related, and leukemia is bone cancer, bone marrow cancer, and lymphoma and myeloma comes under immune system cancers. So treatments includes many therapies, chemotherapy, and now immunotherapy is also there, radiation. So it depends on the nature of the cancer, which is inside the patient. So these have adverse effects also, and which, because these work on cell cycle parameters and to stop the mitosis process. And these chemos are very dangerous, and these have lot of side effects. And these are very two side effects I have included here: loss of hair and nausea. And other than this, there are very severe side effects of this. So there is the need to go for alternate treatments. So in the form of nutraceuticals or through plant extracts, phytomedicine. So we are keen on working about Rincosia products. Rincosia products means in the sense flowers, bark, roots. Actually, roots are very famous now. So we are keen on Rincosia family, which comes under Fabaceae. So why natural medicine is requirements? So these are very less side effects, and almost all parts of the plant are used as medicine nowadays. So here leaf has the major role in generating this phytomedicine. Here in my chart it is 26%. So even roots, every part is useful. So we have focused on this plant extracts. And so far, the work has been done on alkaloids, indole alkaloids, which are extracts of leaves. And there are major anti-cancerous agents, the flavonoids, flavones, flavonols in the plants. And among the plants, we have selected this Fabaceae family, so which is a endemic plant in our area. Seshachalam hills is in Andhra Pradesh, and we have got these Rincosia herbs there. So we focused on this. Out of 300 species which are throughout the world, 22 exist in India and also more in AP. And these are herbs, twinning erect shrubs, and there are a lot of investigations earlier on Rincosia herbs, and these are rich in the production of C-glycosyl flavonoids. And example I have quoted here is Rincosia volubilis. So this has been reported recently, having anti-proliferative activity, and Rincosia villosa has tyrosinase enzyme inhibitory activity, and reported by Rondo in 2017. So these are the photographs of Rincosia family, and Rincosia bedomi, Rincosia minima, and Rincosia bracteata. And actually, the coming to anti cancer activity, so it was showing effect on this liver weight, SGPT, SGOP, ALP, these values. These are taken from research article. And and why we prefer flavonoids means flavonoids are the complex compounds. So these are also integral part of the human diet, and these are present almost in all flowering plants, particularly which we consume. And in vitro work has been done already on this. And these are showing anti-tumor activity, anti-cancer effect, and basically working on cell growth and kinase activity inhibition and apoptosis induction and all. And animal studies are also done on dietary flavonoids and has been reported having anti-tumor activity. And the major thing is hydroxylation pattern of this B-ring of flavones and flavonols. Example is luteolin and quercetin. So this critically influences the apoptosis and protein kinase inhibitory activity and anti-proliferation. So these have the targeting cell surface and signal transduction enzyme activity, such as protein tyrosine and tyrosine and adenosine kinases. So out of all this, Vitexin has been found to be effective. So already a lot of reports are there on mice and human cell lines also. So we have chosen Vitexin. So Vitexin is apigenin 8-CD glucopyranoside. So this is a flavonoid compound which is present in this Crataegus pinnatifida, so in China, and also in Rincosia family also it is found. And this has lot of effect on human cancers, including leukemia and hepatocancers. And aerial parts are having this antioxidant activity, And this has been also detected by DPPH color change from purple to yellow color. And these 5 famous C-glycosyl flavonoids are vitexin, isovitexin, vicinin 2, orientin, and isoorientin. So these 5 are famous anti-cancer agents in the form of C-glycosyl flavonoids. And even DFT, density functional theory, will explain this molecular factors of this, the property of electron distribution. And so we have chosen this vitexin, and it has been already reported about vitexin and isovitexin having effect on different types of cancers: liver, colorectal cancer, oral cancer, leukemia, esophageal, ovary, lung and skin cancer, and breast cancers. So based on this, we have planned in our laboratory. I have not incorporated my data. So we have planned to check on lung cancer cell lines and the effect of this vitexin. We want to extract and study the effect of vitexin and isovitexin. And also there are the reports that Rincosia plants polysaccharides are also helpful in anti-cancer therapies. And we want to maintain all this in our laboratory, the data of all this, including the extracts. And carcinoma cell lines, we are planning to check with vitexin and isovitexin. Yes, ma'am. Yes. No, actually, I have not incorporated my. Yeah. Yes. Yes. No, we are doing. Yes, sir. We have started. We want to work on lung cancer cell lines, non-small cancer cell lines, and we are doing this. So I did not. Ma'am, actually, this is available in our area, and we want to focus on this one, Fabaceae family, and there are the reports already done on the Rincosia herbs. So, yes, ma'am. Yes, ma'am. In the areas near the forest, Seshakulam forest, people are using this for treating. For like, sir, it is like traditional treatment. Sir, actually, we are planning to do this LDH activity in the cell lines. Means when the cell breaks, the LDH will come out. So we want to check that. We want to check this because it will stop this cell growth. No, sir. When cell breaks. Yes, sir. Yes, sir. Rincosia minima. Rincosia minima. Oh, yeah, woman scientist. Yes, ma'am. Yeah, we started, but I did not incorporate the data here. Yeah. Thank you.
Pointer, pointer.
Okay, good morning, everyone. I'm Dr. Akanksha Singh. So I'm currently a postdoc fellow at IGIB South Campus, and I did my PhD from Department of Zoology, University of Delhi. And before starting today's talk, I'd like to thank the organizers and students for organizing this conference so well and for giving us a platform to present our research. So for today's talk, my topic is combating silver nanoparticles mediated in vivo toxicity with curcumin. And in this talk, I'll be talking about how we can harness the potential of phytochemicals and particularly curcumin to combat or ameliorate the toxicity, in vivo toxicity that is mediated by silver nanoparticles using Drosophila model system. So in general, nanomaterials are nano-sized materials which measure less than 100 nanometer in at least one dimension. And based on their physicochemical properties, nanomaterials are divided into several groups, amongst which the metal nanoparticles and carbon-based nanoparticles are most abundantly used. And certain properties of nanomaterials make them highly favorable or advantageous as compared to their bulk counterparts, as these nanomaterials have large surface area to volume ratio so that they can penetrate into smaller spaces. And these nanomaterials, they have superior optical, plasmonic, and electrical properties, which make their incorporation into several industries. And they have a propensity to diffuse across biological membranes, which make their incorporation into biomedical industries very easy. So I've chosen silver nanoparticles for my study because these nanoparticles are most abundantly used in a variety of sectors. So particularly these nanoparticles, they display an antimicrobial potential, and therefore these are used as disinfectants. And in biomedical sector also, silver nanoparticles are used for drug delivery and etc. However, many studies have reported that these nanoparticles display toxicity inside biological systems. And importantly, their physicochemical characteristics such as size, shape, and surface functionality, surface charge, etc., are key or important components in determining their biological response. That is how they would react or how they would behave inside a biological system. Now, it has been established that the major pathway, we can say, or the major route of nanoparticle induced toxicity is through induction of oxidative stress. So once the nanoparticles, they get inside the biological system through dermal absorption, ingestion, or inhalation, once inside the cell, it elicits vast oxidative damage, which ultimately leads to inflammation in the cell and apoptosis of the cells. So why did I choose curcumin for the study? We know that curcumin is a very potent phytochemical which has strong antioxidant, anti-neoplastic, anti-inflammatory properties. And it possesses least side effects in biological systems. So over the decades, curcumin has been vastly studied, and it has been shown that it is well tolerated inside the biological systems. So to approach our question that how we can abrogate AGNP mediated toxicity with curcumin, we chose to take two approaches. In the first approach, we have incorporated AGNPs. We have incorporated curcumin along with AGNPs in the fly food. That is, we have co-supplemented curcumin with AGNPs in fly food. And in the second approach, we have synthesized AGNPs with curcumin extract, and we have fed that to Drosophila to see its in vivo effect. Now, for our first objective, which was to co-supplement curcumin with AGNPs in the food, we had these experimental conditions, that was control, that is DMSO fed, AGNPs fed, and AGNPs plus curcumin fed, and these concentrations were determined post-analysis of dosage, post-dosage assessments. And for our second objective, we chose to study different nanoparticles, that is nanoparticles which are synthesized through different routes, that are chemically derived nanoparticles and aloe-derived nanoparticles, turmeric-derived nanoparticles, and aloe plus turmeric-derived nanoparticles. So we wanted to check what would be the effect of the mode of synthesis on the in vivo effects. And for that, we used Drosophila model system. Now, starting with our first objective, which was to check the effects of curcumin co-supplementation with AGNPs in Drosophila. So earlier in our lab, it has been shown that AGNPs mediate vast systemic toxicity in Drosophila, including detrimental effects were seen on its behavior, its climbing ability, its feeding, and behavior metabolism, as well as at the cellular level as well. So next we checked whether curcumin was able to subjugate this toxicity or manage this toxicity that was seen by AGNP's ingestion. So first we checked the pupation and eclosion rate of the flies, and we found that indeed curcumin was able to rescue the decline in pupation and eclosion of the flies that was mediated by AGNPs. Now, a hallmark manifestation of AGNP's ingestion is loss in the cuticular pigmentation of the flies. And it has been shown that this loss in pigmentation generally correlates with the systemic toxicity in the flies. And we can see that curcumin co-ingestion with AGNPs could partially rescue the loss in pigmentation of the flies that was mediated by AGNPs. And further, we checked its effect on motor ability. It showed an increase, curcumin ingestion with AGNP showed an increase in the motor ability of the flies. However, that effect was not statistically significant. Next, we checked whether the food intake of the flies was altered with curcumin ingestion. So it has been shown that the ingestion of AGNP's food reduced the food intake capacity of the flies at the larval as well as at the adult stages. So interestingly, we found that at the larval stage, the curcumin ingestion with AGNPs was able to rescue the loss in food intake, and these larvae were feeding equivalent to that of the control larvae. However, at the adult stage, we could not find any difference in the AGNPs and AGNPs curcumin fed group in the food intake of these flies. Now, the nutritional status of the larvae determines many of the adult traits. And importantly, the fertility of the flies is affected by the nutritional status of the flies. And therefore, we checked the fertility of the flies, and we found that curcumin was able to assuage or increase the fertility of the flies that was lost by AGNP's ingestion, both at the ovary developmental stage and at the egg laying stage as well. And we followed this till day 30, and we saw an increase in egg laying capacity at day 30 as well. Next, we checked the oxidative stress and apoptosis in the larval tissues to check the systemic toxicity that was mediated by these nanoparticles and further by curcumin. And we know that curcumin possesses excellent antioxidant activity. And we found that curcumin ingestion with AGNPs was able to manage the excessive oxidative stress that was generated by AGNPs ingestion in the larval tissues. So we checked the systemic tissues of fat body and wing disc, because these are the main metabolic tissue and developmental tissue in the flies. Further, the same trend was seen with apoptosis as well. So these flies, when they eclose, they show immense dysregulation of metabolism and behavior, flying, etc., which we think that it might be because of the detrimental effects that were seen in these tissues during the larval stages. So to summarize this finding, we found that AGNP's ingestion, curcumin ingestion along with AGNPs could rescue many of the detrimental effects that were seen at larval stages. So it could reduce the oxidative stress and apoptosis in the several tissues of larvae, and it could increase the food intake and pupation rate of the larvae. Further, at the adult stage, it could rescue the melanization loss, that is the pigmentation loss that was seen in the flies, and it was able to rescue the ovary development and fertility of the flies. Next, we saw the effects of biosynthesized AGNPs, that are AGNPs which were synthesized with the plant extracts, that is turmeric and aloe vera extracts. And first we check the pigmentation of these flies, because as I told just now, that pigmentation is a good readout of the systemic toxicity of the flies when fed with AGNPs. So the first panel shows the 25 mg per liter concentration of AGNPs feeding. The second one is 50 mg per liter, and the third is just the panel to show the turmeric response. So AGC refers to the chemically synthesized AGNPs. AGBT refers to turmeric derived AGNPs, and AGBA showed toxicity, increased toxicity, and therefore we did not find any flies at 50 mg per liter. However, you can see here AGBA refers to aloe derived AGNPs, and BAT refers to aloe plus turmeric derived AGNPs. So these AGNPs were given to us by our collaborators. They synthesized it and extensively characterized it with XRD, FESM, TEM, etc. And we tested the in vivo effects of these AGNPs. And we found that at 25 mg per liter, turmeric was showing a superior response, and the same was seen at 50 mg per liter. And interestingly, at a very high concentration of 250 mg per liter only, we could see a slight reduction in the pigmentation of these flies, which was telling us that turmeric derived AGNPs was best tolerated in these flies. So next we check the pupation and eclosion rate. Yeah. So again, we found a similar trend wherein the AGBT or turmeric derived AGNPs fared the best. That is, the flies that were eclosing from these groups were comparable to the control group at a lower dose at 25 mg per liter. Again, the survivorship and climbing ability of the flies were also the best with turmeric derived AGNPs. Then we check the fertility or the egg laying or ovary development of these flies at day 10 and day 30. And we found that so it has been shown that turmeric has a positive effect on fecundity of the flies. And we found that the AGBT group did not show the toxic effects on ovary development that was seen with other nanoparticles. And only at day 30 at a very high concentration, we could see a decline in fertility or ovary development. So to summarize this finding, we found that the turmeric derived AGNPs, so it is not that all green synthesized AGNPs would fare better in vivo, and it actually depends on the properties, the physicochemical properties and in vivo interaction of these nanoparticles. So we found that turmeric derived nanoparticles were particularly suitable for their application in vivo. So this was confirmed by both the when it was co-supplemented in the food and when it was synthesized with for AGNPs. So these results have been published in these articles, and we have also contributed to this book. And finally, I'd like to thank my supervisor, Professor Namita Agrawal, and Professor Prasanna Shah for introducing me to this new field of nanotoxicity. And I'd like to thank all the lab members present and past, and my friends from the department for their companionship. Thank you.
Akanksha, when you
Yes.
say curcumin derived nanoparticles, what is it you essentially mean?
So basically, they have taken the roots of turmeric. So this is turmeric derived. So they have taken the roots of turmeric, they have made an extract and they've mixed with AgNO3 solution. So that is the precursor solution. So that extract was used to synthesize AGNPs. So this
No, but
is,
what will that extract, what role will that extract have to play
huh,
in the synthesis of the silver nanoparticles itself? Because nanoparticles are generated out of some chemistry that you would be using. no?
yeah.
So what is the contribution of curcumin in that or extract in that?
So in the precursor solution, always a coating, like biofunctionalizing agent is added. So that can either be PVP when when the AGNP is chemically synthesized, it is PVP or some other chemicals. However, in biosynthesized AGNPs, that are the green nanoparticles, these plant extracts are used so that I think they are involved in coating. I'm not very sure of the synthesis part.
Because showing in the difference between the two, when you are extraneously adding the turmeric and then you are synthesizing.
Yeah.
So exactly, we're not able to make out what is the difference. I think you should just see out what is the difference, and
Okay.
anyone else wants to ask?
So we found that by adding curcumin, like it was better if we extracted AGNPs with turmeric. So that showed a much superior response as compared to adding curcumin to the food. So that was one.
Did you go in
difference.
for some molecular studies?
Sorry?
Did you go in for some molecular studies?
No.
Okay,
Not-
thank you.
Uh, the green nanoparticles.
Your nanoparticle
Huh.
are soluble in which solvent, organic or aqueous solvent?
No, aqueous only. They're soluble in water.
There is a problem, it gets aggregated
Yeah.
and precipitated.
Yeah.
What is the
So we sonicate these nanoparticles as soon as we receive them. And then only when they form a homogeneous mixture, then we add to the food.
after sonication there is another problem. The no phytochemicals removed from the have you performed the same analysis before
Yeah.
sonication and after
No, I don't think before and after have been performed. So before only it has been performed, because as I said that these were synthesized by our collaborators and given to us. Then we sonicated them, formed a homogeneous mixture, and so we performed these experiments very fast. And for every experiment, we tried to get a fresh batch of these nanoparticles. Thank you.
On behalf of INCT 2022, I extend my sincere thanks to our chairpersons. Thank you, ma'am. Thank you, sir. Thank you all.